Okay, hello everyone. Thank you so much for joining today for the AIAA Los Angeles, Las Vegas section Saturday meeting. We'll have a very interesting topic today, not only for space biomedicine, but also for commercialization in space, which is a very important subject for NASA. It's not small business, and we also have this is related to the local workforce development and also business as well. So I'm so glad to see Dr. Mename because Aerospace Corporation has a lot of interest that in the commercialization in space. So which this is a good opportunity for everybody to know each other, to get connected each other. So before we start the program, just a few words. We think the library, artificial library, you can see this is a beautiful room. We took us a lot of time to get the spot here. So we try to keep the room clean. So no food in the room. Bottled water is okay. So if you have trash, please bring to the trash can outside. The restroom, you walk outside just across the door. And other than that, I think we are pretty good, all set. Then a few words about AIAA. AIAA is a nonprofit professional organization. As I chatted with Ms. Shepherd earlier before the meeting, AIAA came from the merger in 1962 with two distinguished organizations. One was with the Wright brothers on aircraft aviation. The other one was founded by Robert Goddard. That's associated with American rocketry, as you know. So traditionally, those two organizations both founded in 1920s. So they were either in aircraft or rocketry. So the traditional AIAA folks were mostly either rocket or aircraft. The reason why I mention this because today's topic is space biomedicine. Quite some people think that AIAA might not have something to do with it, but actually it's not. AIAA actually has a very strong group of technical committees. Have biomedicine, microgravity. Personally, I'm involved with microgravity space processing technical committee, and the space medicine has a lot of things to do with human space flight. And actually, AIAA has a lot of people involved with this issue, but traditionally, people think AIAA as either rocket or aircraft. That's just the way it was. But AIAA certainly has been trying to build a connection with different industries. For example, Alina, later you'll hear her presentation. She actually presented in the AIAA Ascent National Conference in Las Vegas in October, right? Later October. So that when she and I met, so arranged a local chapter because anything, just like we chatted with Deborah, we'll have Deborah introduce herself later. Is that anything you come down to execution of any policy or effort, you come down to the local chapter. That's what we are. And locally, AIAA has 1,000 members. And our mail list sending out to over 9,000 people. Social media have more than 16,000 people. So local chapter is very effective in connecting to the local people and organization. But national people, they will be able to fix on something bigger, and they have more resources like Ascend to push forward some of the national agenda. So just a little history about the space medicine. So honestly speaking, the section, which I mean Los Angeles and the Las Vegas section, which include Long Beach area as well. We have been doing several space medicine events. I can remember in 2017-18, we have a group from Loma Linda University, which remind me, they actually have been doing something similar to SpaceBox, but for academic. And they are not focused on mini labs, they're focused on regular payload, and they have SpaceX launch their experiment. I invited the dean and the main professor to join us today, but unfortunately they are very busy. But some of the attendee might be the student or their staff member from Loma Linda. They also work with like Stanford University. So almost each year we'll have one or two space medicine related event. Actually, last year I was talking to a professor in Belgium. She is the delegate for Belgium to United Nations on this kind of space medicine, astronaut safety, those kind of things. So we are trying to build this, and also for the commercialization in space, workforce development. So this event is very much involved in this kind of thing. Just try to explain that for commercialized space, for AIAA is not a lobbying organization, but we do focus on helping people for space, like policy, public policy, and the workforce development. So commercialization, startup, just like SpaceBox, this thing is related to our workforce development effort and public policy. That's what I want to point out. And also we have a lot of technical community. I chatted this with Bridget, right? Bridget and I chatted a lot. And also Miss Shepherd here, which is highly related. So Bridget and Imani here, and mostly related to helping this startup business, but Ms. Shepherd is involved in the national effort for workforce. So those are the people you might want to get acquainted with. Okay, so if any question, please let me know anytime. So without further ado, we'll start the presentation, because I know for Aeroslab the time is Europe is pretty late. So let me start with today's program. So basically, we will have 3 speakers today. Kimberly, the 4th speaker, unfortunately, she is not feeling very well, but she and a group of her friends will be joining, just attending online. Then she will be giving a talk maybe later on. So the first speaker today is the founder and CEO of SpaceBox Scientific, Mr. Yaroslav Zablatinoko. So he will explain a little bit more about this company and also himself. But basically, he has a strong expertise in molecular biology and genetics. And the thing, as I mentioned, the reason why I mentioned the story about AIAA is related to his background, because he is working in between the aerospace and the biomedicine communities. And he's trying to work out the automated life science mini lab for scalable microgravity research. So let's welcome Yaroslav.
Yeah, thank you so much, Ken, for this introduction. Good morning, everyone. And I'm just going to start right from the slides. I hope you can see them right now. Yes. Can you see it?
Yes.
Perfect. Yes. Thank you. Thank you. Thank you so much. So I started this company, SpaceBox Scientific, as a molecular biologist, and I've teamed up with professionals in aerospace engineering and space biologists who already send their experiments into space to actually figure out how to bring microgravity biology to a completely new level. And the new level is translational research. So basically what we are doing in SpaceBox is we're helping to develop biological solutions that will be used to actually treat patients or to bring real products to the market. Essentially, our mission is to allow anyone from pharmaceutical company to even high school to do biotech research in space. And in SpaceBox, we harness this potential of microgravity for groundbreaking discoveries in drug design, disease modeling, with our user-friendly and automated mini labs. So I will just start quickly with overall introduction to commercial biomedical research in space, what has been already done. And before, one of the biggest companies that has been visiting the space and one of the most important use cases is crystallization of proteins that was done by Merck. They were first who actually found out that proteins in space are crystallized larger and more uniform in size. And this allowed them to create a new drug called Keytruda or pembrolizumab. This is like an antibody that helps to treat the cancer patients. So on Earth, it was not possible to crystallize this protein properly, but in space, it was possible to do just in 2 experiments that they sent in 2017. In 2018, they already got the product. And since this time, it brought more than $7 billion of revenue for the company. Second is Sanofi. This follow up with their groundbreaking research in antibody crystallization. So it has a huge potential to enhance the efficiency and storage of antibody-based drugs. And Lambda Vision is one of the also companies who came to this from slightly different perspectives. So they investigated the production of retinal implants in space, and they're aiming to restore the vision of those who are affected by retinal degenerative disorders. And those are the companies that have been making the most input. Of course, there is also AstraZeneca, Microgravity, who are dealing with cancer biology and doing this in microgravity research. So we are focusing in SpaceBox on 3 main value propositions. First is enhanced drug testing. So in microgravity, when you go to microgravity, you can basically create the disease models of the new generation. So when you work with cells on Earth, it's harder to get them to, let's say, 3D structure. When you go to space, you can actually create the so-called 3D aggregates. So when you work with organoids or spheroids of different disease models, such as cancer or neurodegenerative disorder, it's much easier to do it in microgravity because cells do not pull on each other, do not push, and this allows more complex structure. And more complex means closer to actual human physiology. And when you test drugs on these samples, you can actually breach the gap between preclinical and clinical studies. You can increase the efficiency of your drug testing. Of course, second big value proposition is the drug discovery of proteins, so protein crystallization that I already mentioned. And the third is very important for fundamental sciences. In longevity especially, it's accelerated aging. So when you go into space, the first research was basically devoted to it, was basically, it allowed us to know that in space, the age-related disorders are, you can basically investigate them 10 times faster because of the aging process. Aging is accelerated in space. So each time you bring your experiment into space, you can fast track the experiment and get the results much, much faster. And of course, time means a lot of resources. So in SpaceBox, we are focusing on 3D cell culturing, as I mentioned, so creating new kind of complex disease models, protein crystallization, and from 2028, we're going to go into space biomanufacturing market. So we are focusing on space biopharma market, and this is like a $3 billion opportunity right now. There is also a bigger market that is space biotech. It's focused not only on pharmaceutical compounds, but also cosmetics and food production and all of the stuff that is related to biology in space. And if you go to overall biopharma R&D, which is the global market, it is around $300 billion. So this is a growing market, and there are more and more use cases each year arising. So there are also mRNA technologies that are being done in space, new sequencing technologies, new cancer treatment approaches. So there are more and more use cases each year, and this allows a steady and substantial growth of this market. So in space, cell biotech is growing, yeah, I can say the same, with the same compound annual growth rate as the space biopharma. And in 2030, it will reach $10 billion. So what we identified during our journey in a space biotech, that usually when researchers go into space, they have several different problems. And most of them are, of course, related to the high price, low cost efficiency of the biotech research on the International Space Station. Because NASA changed its pricing policy in 2019 from complete grant coverage to the point that companies have to pay themselves for each research, the prices went up, and still companies have to pay themselves for the whole commercial research. And our mission is to drop this price to allow more companies to go into space. Also, as we've spoken to many researchers worldwide, they all say that sometimes astronauts fail their experiments. So because they have limited crew time or skills, they cannot do very precise, highly skilled protocols that require a lot of time and a lot of attention. So what we do in SpaceBox, we propose one solution, the fully automated and portable mini labs for microgravity research. And this allows us to be not only 3 times cheaper than the competitors because of the automation and portability of our technology, but also to provide the reproducible data with higher quality, because we cross out the human factor and automate the whole experiment. Our portable devices can also be used not only in space applications, but on Earth in clinics. And we developed the machine learning tool for optimization of different protocols. So what I want to actually focus on in this presentation, in this talk, is the how we facilitate the drug testing and how we increase actually the efficiency of preclinical studies. Overall, space biopharmacy and pharmacology is very complicated field, because you have a lot of compounds that influence your experiment, and the drug discovery usually takes up to 10 years and up from $3 billion to $7-8 billion for just one drug to be developed. When we talk about the cancer treatment or neurodegenerative disorders, it's getting more and more harder because of the absence of alternative treatments, and we don't have so much data to build on. So this is when we step in and say that we can increase actually the preclinical study efficiency. So right now it's just 10% of drugs that are tested on preclinical stage that can actually go to approval for clinical trials and then the FDA approval. So just 10% get to the market. And with our technology of microgravity, both microgravity, microfluidics, for increasing the complexity of disease models, we can actually increase the success rate of preclinical studies. We can increase the efficiency of drug testing. We are getting to this point due to simplification of 3D cell aggregation. So it's easier to get more complex organoids or assembloids of different cells. I hope I don't overwhelm you with biology. It's just how the 3D structures of cell formate. So when we speak about just the 2D cell, is just a plate cell in 2D structure. When we talk about the 3D cell structures, it's basically an more comprehensive cell-to-cell interaction, like spheroids. And if we go even further, we can get to the organoid structure when we have a small, tiny piece of the organ that resembles the physiology. As we go further, we can increase actually the complexity from organoids to assembloids and assemble different systems in one, different organ systems in one. So this is what we are targeting with our microfluidic systems, to help to actually get to this point when we can simulate and create the real disease models to test the drugs on them. So we enhance the complexity and size of the models with microgravity and microfluidics. Our autonomous mini labs can actually be used for space and delivered via different approaches. So you can go either to International Space Station, or you can use the free flyers. To get to International Space Station, you can use the docking station. So our technology is compatible with all of the service providers. We also develop our own CubeSat technologies. So we have the manufacturing facility for this, also testing of the payloads, so vibrational tests, safety tests. And then you can also go for different free flyers as Space Riders, the European Space Agency initiative, and re-entry capsules. They have their own benefits, so pros and cons. As we are compatible with all of them, we can get your research from long-term to short-term. You can go for cargo return, or you can basically investigate your research remotely. With CubeSat, you can go up to 3 years in space, but you cannot get your cargo return on Earth as of now. Of course, there are technologies that are aiming for it, that are being developed, but as of now, it's not possible. But what you can actually do, you can investigate for example, one of the research that I have been doing in my studies, I've been investigating how ecology of microbes and plants is being established in microgravity, and we did it on CubeSat. So it was a project of sending the plants into space on CubeSat, and then just do the phenotyping and obtain the data remotely. When you speak about the cells, of course, you want to do some more complicated downstream analysis. You want to get them back. You want to use them for pharmaceutical experiments. And this is when you have to go to Space Rider, re-entry capsules, or the International Space Station. Unfortunately, the International Space Station is still very hard. You have to wait at least 1 year. Sometimes your flight is being delayed. Sometimes you have cancellations of flights. So it's very unpredictable. So basically, the SpaceBox has formed itself as a research institute project. So we are a spin-off of many different institutions that have been all the consortium of Ukrainian Space Agency. So we've been all in the early research in space biotechnology. And then we formed different entities that are providing CubeSats, providing biological research separately, and mostly we've been doing the CubeSat experiments. In 2014, we sent successfully the first Ukrainian nanosatellite, Polyton first. It's still orbiting the Earth for 9 years, which is the global record for orbital duration. In 2017, we released the second CubeSat from the International Space Station to study the thermosphere of Earth. And in 2023, it was the latest project of ours for just thermal simulation inside the CubeSat to carry out these 37 degrees temperatures that we need for cells to be cultivated. And now we have the whole set of technology to send the biological experiment into space, which is our next step. So we have done 3 successful space missions, secured already long-term customers who are willing to go with us into space and also do clinical studies with our technology, secured partners for logistics, certification, and launch. So we have the whole, let's say, end-to-end market for this, end-to-end pipeline. So we can get your idea, develop it, and send it into space. And we have in-house R&D facility for cutting-edge devices, to build cutting-edge devices for biology. We are currently developing it. It's just the first steps that we have, but as we grow, we are developing it more and more, and basically aiming to also get our own preclinical studies on Earth. This is our core team. So people who are doing cell biology and genetics, who've been working in space biotech already, have PhD degrees and working with complex disease models, both in neurosciences, cancer biology. We have people who are representing us from business side, sales and marketing experts, people who are using the aerospace engineering as a stepping point and basically develop not only nanosatellites, but also propulsion system, telemetry solutions. So everything you need to get actually data back from space to Earth. And of course, I'm grateful for our scientific advisory board, people who've been sending the experiments into space. Dr. Alison Nuotri, who sent organoids into space this year, actually recently, and many other people from Yale University, Czech Republic, and Ukraine. So together, we open affordable access to translational research, helping to improve billions of lives of people on Earth through microgravity studies. Thank you so much. I will be happy to answer any questions, so please feel free to jump in with the question.
Yeah, I have a question. This is Martin McLaughlin. Can any of this be done suborbitally, or do you need months? Can any of it be done if, say, you had 15 minutes of microgravity?
Yes. Many experiments can be induced with altered gravity, with a zero G flights, for example. You have 15 minutes of microgravity, but this is not enough to study, for example, the time lapse of how 3D aggregates are forming in space. So, for example, the microqueen, they want to see how the breast cancer is being developed in space from day 1 till day 90, 160, 300. So they want long-term studies in space. And this is when we can provide it with the CubeSat technology. You can go into space for 3 years, for example. Many plant sciences studies also require very long time because of the plant physiology is quite slow. The metabolism is also slow. So you have to develop some long-term approaches. And when we talk about protein crystallization, it's not possible to do it in just 15 minutes. So we have to go to space. We have to be present.
And I have a follow-up question. How sensitive is it to the reentry environment? Would a lot of this, is there a great value in having a gentle, soft reentry, or can a lot of these experiments take, a typical, capsule-like hard, 6G reentry?
Yeah, this is a big issue for us. So the only possibility to actually address this issue is to preserve your cells before the flight. And when you reach the low Earth orbit, you have to somehow, via temperature or chemical adaptation, you have to defreeze it, introduce it into the culture them, freeze them, or fixate them, and then bring them back to Earth. To have a very clear experiment when you have a really good control of microgravity research on Earth. So either through orbital, RPM machines that just circulate your experiment, and then on Earth in space you have the actual research when you have only Earth so LEO, the low Earth orbit microgravity exposure. So this is
both the launch and reentry environment is an issue for you.
Yes, this is the issue, but only if you don't preserve your cells. So at first they experience, of course, the hypergravity that leads to some of the physiological and biochemical responses. But if you can preserve your cells, it's a good way to do it.
Thank you.
Yeah, thank you. It was a great question, actually.
I think Richard raised hand. Richard, go ahead.
Thank you. I have so many questions. I've been part of 6 different stem cell experiments to ISS. Actually, seven. 6 on ISS through shuttle, and 1 through CRS-20. And I will definitely be wanting to talk to you, Yaroslav, more in the future. I guess the main question I have is, are you a technological facilitator, or do you actually run some of these experiments yourself with a view to developing a biological product that is saleable?
Yeah, this is a great question. Thank you, Richard. I will be happy to also keep in touch in future. And to answer your question, Currently, our business model is to create only the facilities for microgravity research for biologists to use them. And later on, as we grow, we aiming to establish our own R&D facilities on commercial space stations to do our own research to help pharmaceutical companies to develop their drugs, for example, and then get the revenues from the drugs themselves. But for now, it's not very feasible business model. So we are just focusing on, the services.
Okay. Yeah, because I spent 2 years working on Dragon Lab at SpaceX, and a lot of what we were doing was looking at all the different types of payloads that we would put up on a free-flying microgravity commercial lab that didn't go to station. So it had a little bit more wiggle room where, you know, we didn't have to jump through hoops, NASA hoops to get on station, which is substantial. And to the previous gentleman's question, the G load on a Dragon or a Soyuz is fairly substantial. That's why we're looking forward to the Dream Chaser having a little bit more of a benign G load on the way back, because yes, it does tend to have a deleterious effect on delicate cells. Were you aware of some of the work that was done on the last Columbia flight by Dave Wolf and his team when they were growing cancer? They were trying to grow in vitro prostate tumors in a revolving wall bioreactor on the ground as a control, and then they had one on orbit on the Columbia. The ones on the ground, they were able to develop these kind of a lot of microblastules about less than a millimeter across, but on station or on the shuttle, they were able to create a 15 centimeter tumor. It's a 15 centimeter long tumor. That's in the record from that flight. And also, I noticed you said Merck had done some of the protein crystal growth stuff, and it was, I think, some of what you might have mentioned was done by Corning before they were acquired by Merck, and that was an experiment that was on. It was an interferon experiment, protein crystal growth on Columbia, that survived the crash to the ground, and they were able to take that out of solution from the recovered experiment that crashed in Texas, and that's what I believe was used to make a hepatitis C vaccine. But NASA will never talk about it because it's proprietary information. And so, you know, when we ask why are astronauts risking their lives, it's because of that. But NASA's not allowed to speak about it. So there's a lot of really cool science that's been done, and there's so much more that we need to do, and I'm very much in favor of what you're doing. I think it's brilliant. It's fantastic. I guess what my last question, apologies, what's your footprint in the United States, and who are your investors?
Yes, okay. Currently, we are supported by grants, and we are representing mostly, we have the office only in Germany, and we are, basically aiming to also establish our own office in the US. And we just have people who are supervising us from the US, and people who are part of our team. So for example, Alina Voronina, she's here, she's our main business developer. And as we grow, we want to go into the US as well. It was a great point that you mentioned. Thank you so much. I'm really interested in the prostate cancer experiment. Did they have the good viability inside the 3D cell aggregate? Because one of the issues is like, yes, you can get a good spheroid that is complex and is big, and it resembles overall the same like cells that can be found also in organ, but the cells inside and the transcriptomic level, so how the cells communicate between each other, is very different from the real model. So what is actually we are aiming to do is to preserve the cells that are inside the aggregates that have less nutrients, less oxygen supply, to not die out and have a good communication with outer layers of the 3D cell aggregate. And this is like a stepping point. This is like a stepstone that we are aiming to change with our microfluidic systems. This is a very good point.
Thank you. Yep.
Okay, thank you, Richard. Alondra, I think you are online. Dr. Albrecht, do you want to speak up?
Yes, hi, thank you. Can you hear me?
Yes.
Okay, hi. This is fascinating research. I have very limited understanding of space medicine, some background in biomedical research, but I was just wondering if different types of space, cosmic radiation can affect the results that you're assessing with your microgravity experiments. It would seem certainly for biological agents that might be more relevant, but I don't know what the research looks like in terms of evaluating microgravity and radiation in concert. I know that they are kind of the two biggies when people talk about human survival in space. So I was just wondering if you could shed any light on that.
Yeah, thank you so much. It's a great question, actually. It's a question that I think Davon can answer better than me. Basically, you have different exposures of radiation. So I think it will be a part of the Davon presentation as well. I don't want to dive into depths into it. He will cover it better. But when you are present on the low Earth orbit, you have you don't have such a high radiation exposure inside the International Space Station. So it's physiologically our physiology can withstand it. So we don't see any substantial increasement in the mutations. Correct me if I'm wrong. If we are on the low Earth orbit and we have the protection of the from the radiation, so we have some radiational shield. But if you go into outer space, if you go to the moon, Mars, or deep space, you have less and less protection from the radiation, and then it certainly increases the mutational rate, the amount of chromosomic aberrations, and all of the predispositions to cancer disorders and many more disorders that are related to radiation. So this is a good point. But on the low Earth orbit, it's not that harsh, the radiational influence, especially on the 3D cell culture.
Thank you.
Okay. Very good. So, Yaroslav, can you stay a little bit longer? We are about to go to sleep.
Sorry, I didn't hear the last sentence.
No, I mean, can you stay a little bit longer, or you have to go?
Yeah, I will be happy to stay for the next talk, but unfortunately, then I have to leave because I have my flight soon.
Yeah, because I think because Alina is here and for your company, for a whole picture, it's better after she present her talk, so then people can have the whole picture for the operation here. So Alina, so you're ready? So let me get your file. So have you emailed? We have a link. Oh, very good. You can type it here. Go ahead.
I sent it to your email.
Oh, sorry.
It's okay.
All right. Oh, because people online cannot see. So we have to share your share. Can you see it? Let me see.
Yes, perfect.
You can see it, right? Okay, very good. Let's see. Now, if you do this, Well, I guess it's fine. You just have to minimize this.
Mhm.
So you just, if you need, like a laser pointer or something?
Oh, fine.
Okay.
Thank you. Thank you, everyone. Firstly, good morning. Then thank you, everyone, for coming here this morning and dedicating your time to listening to our lecture and exploring more about our company. And thank you, Kim, for letting us to do this lecture. It's our kind of first experience for doing a lecture at the university. So let's get started. My presentation is going to be different from Yaroslav because I'm going to be elaborating not just about our company, but just importance of biomedical research in space for anyone who is not probably in biomedical field or who is entrepreneur, startup developer who are just interested explore more about this topic and learn why it's important to do this research in low Earth orbit. Thank you. I'm gonna start with my career. So I started career as a scientist at 12 when I wrote my first scientific work, and then I became the member of Junior Academy of Sciences of Ukraine at 13 years old. Currently, I'm 17, and I recently became scientist at New York Academy of Sciences, and I'm also Chief Business Development Officer at SpaceBox. So I'm graduating from a high school here in Los Angeles, and I moved from Ukraine 3 years ago. And it's an honor for me to be a part of SpaceBox because we already made a lot of improvements. And the first picture, you can see me participating in his challenge on ASCAN conference, where we met with Kim. And then on the second picture, and the third picture, it's what I'm doing besides SpaceBox or what I was doing. So that's me, a speaker also in Central Asia. I was speaking about my third scientific work, which was dedicated to energy recuperation equipment. And on the middle picture is me with Deputy Minister of Education and Science of Ukraine, who was, I was honored to meet him because of my career, and he really wanted to talk to me and say thank you for everything that I'm doing. And right now, we're gonna dive into importance of biomedical research. So there are tons of things that you can actually do with technology that we are introducing in SpaceBox. I'm just gonna name some of them, but that's also not a limit. So for example, protein crystallization, which Yaroslav talked about, cell cultures, microbial behavior, drug delivery system, immune system, pharmacodynamics, cancer research, drug screening and testing, molecular biology, and gene expression. And on the picture, you can also see what biology you can do with lab-on-chip technology, which I'm going to talk later on, and the chemistry. And here you can see a picture of process of protein crystallization. So lab-on-chip technology is the main technology that we use at SpaceBox, which allows us to research all of those topics and things that I counted on. It was introduced in the 80s in the US, and researchers made a concept of lab-on-chip in order to make a revolutionary diagnostic tool. Firstly, it was created for manipulation with small volumes of fluids and liquids, but later on it was introduced for drug development. These complex systems can be adapted for space missions to conduct a variety of experiments in microgravity conditions, facilitating research across multiple areas related to drug development. Lab-on-chip is a dream to integrate into a single chip thousands of biomedical operations using a single drop of blood from the patients to precisely diagnose potential diseases. Lab-on-chip provides the whole new world for opportunities of DNA and RNA sequencing. The first human genome projects took years and required hundreds of scientists of their work. Today, by integrating an array of DNA probes into lab-on-chip, we can sequence genomes thousands of times faster. Moreover, nanopore technologies, which is still needed to be optimized, hold great potential in the future for being such faster for genome sequencing. Platforms are being developed for testing long and short fragment gene detections. single base substitutions and intersections, and pathogen-specific DNA sequences. It will take a long time to describe all currently ongoing research of nano microfluidic lab or lab on chip technology. It's enough to say the contemporary research lab on chip technology focuses on three main aspects. The commercialization of lab on chip, including the adaptation of fabrication processes, the design of specific service treatments, and flow control systems. The second one is parallelization of the maximum number of biological operations integrated on the same chip. And the third one, fundamental research on high potential impact technologies, such as DNA reading through nanopores. And then I'm gonna talk about how actually we can make this research happen. So we can test our experiment or hypothesis on traditional microgravity platforms, including drop towers, parabolic flights, sounding rockets, short duration orbital platforms, and long duration orbital platforms. I'm going to briefly talk to you about each of them. So the first one is parabolic flights and sounding rockets. Parabolic flights reach an altitude of at least 3 kilometers and provide microgravity for up to 25 seconds. And of course, it has parabolic trajectory. Sounding rockets. A two-stage sounding rocket can achieve peak altitudes over 400 kilometers to attain for 5 to 6 minutes in microgravity, level of 10 in the power of -4 G. Drop towers and random positioning machines. The drop towers are vertical structures that allow free fall of payloads to generate microgravity conditions, the duration of which is determined by the tower height. The 10 to power of -3 G microgravity level that could initially be achieved has been improved to the current level of 10 in the power of -5 G of the most drop towers using techniques to counter the effect of acceleration. The random positioning machine is a two-axis version of a clinostat, which has been used for microgravity simulation and hardware testing. These machines randomly change the orientation of the experiment, effectively averaging out the gravitational pullover. The microgravity is in range of 10 to the power of -2 to 10 to the power of -3 G. Experiments, the statistics of experiments on ISS is truly fascinating. So just I'm gonna read many of the countries who made some kind of biomedical research on ISS. The International Space Station is the largest scientific technological international cooperative program worldwide. The ISS is based on the partnership between USA, NASA, Canada, Canadian Space Agency, European countries, the European Space Agency, Russia, Roscosmos, and Japan Aerospace Exploration Agency. For more than 18 years, over 230 people from 18 countries have lived and worked continuously on board of ISS, conducting 2,400 research projects. Over 200 new experiments will be launched in 2019, and more than 1,200 microgravity-related patents were granted between 1981 and 2017, which is truly impressive because that shows that people actually care about microgravity research and experiments on low Earth orbit. And of course, there is research being done from private companies who are also in a partnership with ISS, which are Space Tango. So they do Tango Labs that we can take, that I will talk later on, and space application services with Ice Cubes. Usually they provide services of 1U CubeSat models, which allow us to conduct, not just test a hypothesis, but conduct a whole research on low Earth orbit. And here you can see 1U CubeSat of Space Tango. Those are microgravity platforms that allow us, as I said, not just to test, but to also conduct an experiment. And in SpaceBox, we have a specialty in two of those groups, which are satellites and free flyers. And you can also see different types of satellites, which are biomedical research, are mostly done on CubeSat, but of course it depends on complexity of research. And I want to talk about two space missions which are truly fascinating from my point. One is space mission done by Space Tango, and it's one of our advisors, Dr. Alison Melchi, that already Yaroslav talked about. He done organoid research with Eric Viret, studied human-brain organoids in space. This mission was done in 2019, and recently this year they done another mission on organoid brain research, but they didn't get data on it. And the second experience that I want to talk about is Keytruda Barmork, which Yaroslav elaborated on. And I think Yaroslav said everything that you need to know about it. So they crystallize the protein, which is used to treat several types of cancer. And they just show the limit of the potential that research on low Earth orbit has, because they generated 7 billion in revenue, which is they totally commercialized the whole biomedical market. I hope you enjoyed and explored something new for yourself. And this is how I was explained when I came to the company from Yaroslav. So I made it very easy so anyone can understand, from a physicist to life scientist. And thank you so much. If you have any questions, please ask.
Thank you, Alina. This great presentation. So what did you present in Ascend? Is a similar topic, or did you say something more in Ascend?
I said mostly everything that Yaroslav said, so it was mostly about the company. Oh. It was 5-minute pitch about our company and what services do we provide. But this is just a generalized presentation about what company do.
So any question for folks, people, or online? So here,
I have a quick question. You're showing a mix of human-tended and automated experiments. Do you have any? How much value does the human-tended add to it, if any?
Sorry, can you repeat your question? Because I think because of the internet, I couldn't hear.
Yeah, sure. You showed a variety of human-tended experiments with an astronaut on board the ISS. I guess most of it, maybe some of it was in the shuttle. Nowadays, how do you see that going in the future? Will we still want to have some human-tended experiments, or can they mostly be automated or ground controlled?
Thank you for this question. It's truly important because right now the main problem of the market is autonomously and the cost. It's too expensive to send an astronaut on the board. And if it would be automated, of course, it would solve a lot of problems. So what we are doing in SpaceBox, we're trying to make our system fully autonomous. And we are hoping there are a lot of companies who are willing to build private space station like Axiom Space, and who would be truly interested in, of course, taking care of our experiment autonomously. So I believe the future in the space is a future of biomedical research. It's autonomous, but of course we can't fully get rid of humans, because there are a lot of complex experiments that require human work.
Can I just jump in? Thank you so much for elaboration, Alina. You say everything that that is important about automation. I think just one small add-on is when we go and think about the future, and we see the NASA roadmap, we see more and more going to deep space. Not only Moon and Mars Mars experiment, but also deep space experiments that basically don't allow currently human space flights. So it certainly won't be carried out by humans. So you need some autonomous approaches to carry out the whole pipeline of experiments from the beginning till the end with automated solutions. And this is something that we are seeing us in the future going more into deep space and exploring more with our automated approaches.
Very good. So Yaroslav, I know you are going to go to space soon, and Alina is here in local. So because I really want to connect you with Bridget. Bridget, are you still online?
Yeah.
Yes, I'm still here.
I had a question about the protein crystal growth side of things. One of the things I found when I was working at SpaceX was that there seemed to be a window of opportunity for protein crystal growth on orbit. The original reasons for growing the protein crystals on orbit is that you could grow bigger protein crystals. They grew much larger. And so therefore, with the ability to image the protein down to 50 or 60 nanometers back then, you couldn't see those smaller proteins like urokinase or interferon. And so they would try and grow them bigger to get them bigger than the 50 nanometers. But then the new X-ray synchrotrons were coming online, and they were getting us down to much, much smaller nanometers in terms of how we could image those proteins. And so I saw that as a kind of a window of opportunity, that now you can get down to those 5 and 6 nanometers. However, you can still grow more pure crystals in space. And I'm wondering if you guys see that as the main killer app, as opposed to just trying to image the shape of the protein.
I think that's mostly a question for Yaroslav, because he's our scientific side.
Yeah, thank you so much. This is a great point, and this is something that we definitely see a large gap. So the purity of the crystals that we can get. I think the previous experiment that was done by Merck and also Sanofi with their antibody, it was kind of cutting edge technology that they used. Definitely, the purity of the protein is important, and we are collaborating mostly with research institutes from Düsseldorf, from Germany, that are doing crystallography on the regular basis, and they have approaches like a hanging drop approach, for example, that gives a nice purity if you get proper conditions for the protein folding. So yeah, the purity is definitely the key point here.
Oh, okay, thank you, Richard. So you're welcome to ask more questions, but since Yaroslav has to leave, so Richard, can you say something about you and what you're doing regarding to this training for startup company?
Sure, yes, happy to. So thank you, Yaroslav and Alina, for that very, very useful presentation. I think where we may be able to help is with the US expansion. So I was overseeing a program with Starburst Aerospace along with UCLA, where we are taking small cohorts of companies, usually 8 to 10 companies, through an accelerator program that is completely free. It is a 13-week program that is aimed at helping you expand your team, connect with the government customer, explore more of the commercial side, and then connect with US investors. We ran the last cohort that we had concluded this summer. So we ran that from May to July. But I know that there's definitely things coming online in the new year, specifically in the space health vertical and exploring more biomedicine. So we'll love to stay in touch with you guys to see maybe how we can support you guys going forward with that.
Definitely. Thank you, Brian, for bringing this up. Yeah, we will be happy to have a follow-up call with you. Thank you.
Absolutely.
Thank you. Yeah, thank you for this proposition. I'm always in LA, happy to, contribute to your program. So thank you.
Yeah, that's fine, Bridget. I was hoping Bridget can be here today. But we also have Amani, seems to be also related to the helping the startup company. Do you want to say a few words?
Oh, this is all very new to me. I work in an investment management firm and follow some of these larger cap pharma companies like Eli Lilly and Merck. And so it's not something they talk about a lot publicly. So this is just so interesting to hear that Keytruda, a very blockbuster drug, was aided by this. So I'm very new to this topic, but I'm really happy to be here and I've learned so much. So thank you so much.
Yeah, very good. So you can also connect each other and see if you can work out. Nothing involved with this kind of commercialization is workforce. So we have Deborah here. So Deborah, do you want to say a few words about what you and about what you were doing?
Well, thank you, and hello, everyone. I'm with the South Bay Workforce Investment Board located in Hawthorne. So now everybody knows what Hawthorne is, thanks to SpaceX. But we've been around for a long time. We're a 501c3, and there's a small group of us that are called special projects. And what we have been doing is following the hot industries, if you will. And space is certainly one of them, along with healthcare and bio, through our what we call our Flex program. So you may have heard of Aeroflex, BioFlex, and now we have SpaceFlex. It really started because the White House I think it was in January 21, the Vice President's office held a convening of a number of space/aerospace companies, for those that were working in space, to find out how they could build the space technical workforce. And they realized that the only way they're going to get anywhere is to go out into the country. And so they established regions, starting with 3, Florida being the first region, Southern California being the second, and the kind of central New Orleans, Texas area as the third, and now they're looking at the Colorado area and the Mid-Atlantic. And what the goal is to bring together companies that that need workforce from a technician level up, and to get together and say, what can we do better together to develop a workforce and a pipeline, and what do we need to do to help education, to help people know about space occupations and career pathways. What can we do as a team that maybe we can't do as well one-on-one? So that's what we are doing. If you are interested in being part of this group, please let us know. We are expanding the group. There were about 10 companies that came to the first convening last March. And then we've had other one-on-one meetings to really get to your needs. And then we are anticipating a second meeting, most likely held at Northrop Grumman in next March.
Excellent. And I know Mr. John Beza online seems to be also, Richard, I'll get back to you. John Beza, you're supposed to be here in person, but you're online. Do you want to say a few words? I see you seem to be also involved with some kind of workforce issues. John, you have the mic. You are welcome to speak out. Mr. President, you want to say something? Or Ms. McCoy? All right, we can get back to you when you are ready if you want to say something. So, I think that's good. So I'm trying to make some connection for you guys. So thank you, Arslan. Thank you, Alina. Appreciate it. Wonderful presentation. Thank you. So we'll be moving to the next talk by Dr. Mundi. Let's see.
Yeah, I'm sorry. Thank you so much. I have to catch my flight in 2 hours, so I have to go. Thank you all. Yeah, let's stay in touch and have a great day. Thank you.
Yeah, thank you, Aristotle. Stay in touch. So let me see, where is, okay, Dr. Lundin is here. Let me for you here. Okay, do you want to put up your PowerPoint?
Yes, can you hear me okay?
Yes.
Okay, I'll just share my screen.
Yeah, share your screen.
And then maybe, I don't know if someone may need to moderate the chat questions at some point.
Yeah, no problem. So the next talk is by Dr. Lundin, Devon Lundin. He is a postdoctoral research fellow in the Memorial Sloan Kettering Cancer Center. He completed training in City University of New York Graduate Center. Then at that time, he examined DNA replication speed in triple negative breast cancers, harboring mutant p53 proteins. Right now, he focused primarily on DNA repair mechanisms, and he's also doing this radio sensitization of HPV plus cancers, and in radio protection for astronauts during long-term spaceflight mission. So go ahead, Dr. Lundin.
Well, thank you very much for a great introduction and for the AIAA for hosting me. Yes, I'm very proud to talk about my project today, modulating the DNA repair response to shield astronauts from cancer. So I was also very fortunate to be at the Ascend meeting in October, and I was able to win the Humans in Space Challenge on the research side, and that was hosted by Boryung, Axiom, and Aurelia. So if you can see me, I've got the Axiom station behind me. So that's my personal favorite for now because they're funding my research. But any company that wants to fund my research, I'm happy to put your space station behind me when I give talks.
It's good, Dr. Lundin. Can you close? I think there is a panel in the.
Oh, sure.
Because it's blocking your slides.
Sure. Sorry about that. How's that?
No, there's one in the middle too.
Oh, well, it should go away. Yep, there we go.
So you are working
in
Korea. You're working in this Korean company.
Oh, yes. A private equity group has helped to fund the project that I'm very excited to share with you.
Very good. They were in a sense. So Alina and I. Very good.
Yes, I'm a very big fan. So let's see. You may have heard the universe is expanding, and that's true both in the physical sense and in the sense of space research and development, right? So you all probably know, since you're here, about the Artemis missions. Artemis 1 flew in 2022. Hopefully Artemis 2 flies in 2024. We should have an Artemis 3 crewed lunar landing, hopefully no earlier than 2025, but hopefully not too much later either. And then we've got plans, we as in NASA, humanity, have plans for Artemis 4 and beyond to have a sustained lunar presence throughout the 2030s. Beyond that, there's plenty of American commercial space stations which are planned. So the International Space Station has been great to us and is entering the final phase of its life. So it's set to be decommissioned sometime in the 2030s. And we've got all kinds of replacements in theory lined up. Orbital Reef, Axiom Station, as I mentioned, my personal favorite, several others, Northrop Grumman, Starlab, and of course, the Lunar Gateway, which is intended as part of the Artemis missions to help our presence around the moon. We also just launched at the end of October a mission to Psyche, NASA's first mission to a mostly metallic asteroid, and this asteroid is allegedly worth $10 quintillion. So I'm not an economist, I'm a scientist, as you'll see. So I wanted to include that valuation's from Business Insider, and as far as I understand it, that is basically more than the entire global GDP. So I'm sure that number is not really, I don't really know how that works, particularly how supply and demand works, but that's all to say that this is really big business, and we're trying to figure out how to do all these incredible things. And of course, Artemis is meant to be a gateway to Mars, right? And so all of the technology development going towards this is for both unmanned and manned missions to Mars. Since 1957, we see an increase in global spacecraft launch attempts, which includes kind of a release of satellites. And this data goes up to 2021, but 2022 and 2023 were also record years. So we have really big human spaceflight ambitions, but there's a big barrier to all these problems. The fundamental problem, as I see it, is that cosmic radiation exists outside of low Earth orbit, as Yaroslav mentioned, and increases the cancer risk for our astronauts and limits our ability to explore space. So to give you a sense of kind of what this means and a sense of scale, the average American, maybe some of you sitting in the Artesia library, over the course of 12 months, get a radiation dose of about 6 millisieverts. If you go out to, let's say, my favorite, the Axiom Commercial Space Station for 12 months, you get about 20 times that dose, up to about 180 millisieverts. If we succeed in developing the Lunar Gateway, and we have a 12-month mission to Lunar Gateway, you've got 200 to 500 millisieverts that will be experienced by our astronauts. And a Mars mission, which is at a minimum 21 months, probably much longer than 21 months, you'll have a radiation dose somewhere between 400 and 900 millisieverts, depending on the solar activity at the time of the mission. And so these are huge amounts of radiation, and in particular, this 400 to 900 millisievert projected dose is the entire lifetime dose that a human is allowed to experience, essentially, and you'll receive that in less than 2 years. So this is a huge problem. But how does this radiation specifically affect us? There's also, so I'm discussing the quantity of this radiation, but there's also the problem of the quality of the radiation. And so in particular, if you think back to maybe chemistry 1, or whenever you've last looked at the electromagnetic radiation spectrum, maybe this is the first time, you'll see that on the left-hand side, non-ionizing radiation is generally very low energy, right? So things like radio waves, 5G in your cell phone, as much as some people maybe believe otherwise, actually very low energy and does not harm your cells. And on the right side, you have ionizing radiation, much higher energy, right? Including ultraviolet, when you get those sunburns from being out in the LA sun too long. X-rays, whenever you go to the dentist, right? And we usually worry about those as your highest source of radiation as a normal citizen on Earth. Then you've got gamma rays, and all the way on the right, we have cosmic rays. And so each of these numbers in the quantum energy section are increasing by factors of 10, basically. And so an order of magnitude is 10 times more, right? And so the lowest end of X-rays, you've got 10 to the 2, and then on the highest end of cosmic rays, 10 to the 12. So it's many, 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 many more times, about 100,000 times more powerful than those X-rays that you worry about, or the ultraviolet at the beach. So not only is the quantity of radiation much higher in deep space, but the quality of that radiation is also very high. And so what does this do to our cells and our DNA? So we have here pictured in B and C, these are two individual mammalian cells. And so what's pictured is the nucleus, and the green dots are something called gamma H2AX, which is a marker for DNA damage, and in general tells you when your cell is experiencing DNA damage or double strand breaks. And so when cells are hit with a gamma ray, you see these little dots, or something that we call puncta, and it's almost like bouncing ping pong balls off the nucleus of the cell. Maybe they'll leave a little bit of a dent, and it certainly causes damage. But when you look at something like a heavy ion particle, which is much similar to energy as cosmic rays, what you get are these long tracks, linear tracks. So rather than a ping pong ball, it's more like a cannonball going through the nucleus of the cell. And all along the way, as these heavy ions go through the DNA, they're essentially shattering the molecule and creating very complex DNA breaks that are very difficult for the cells to deal with. And so gamma rays, pretty bad. Cosmic rays, really bad. So that's the kind of the takeaway from this slide if you are a less technical person. So how does this relate to increased cancer risk, getting all of this damage to our DNA? Well, this is a a very oversimplified explanation of sort of the theory of cancer and mutation accumulation. But essentially, more mutations equals more cancer. That's not exactly true, but it'll help us in this talk today. And in general, what you see is you need accumulations of mutations in a single cell. So you can get a mutation in a cell, and it can replicate and live its life totally fine with that one mutation, no problem. Or maybe you're out in the sun a little bit too long, or you're on the Axiom space station, you get some cell damage, you get a second mutation, third mutation, a fourth mutation, and beyond. And this is when we start getting into dangerous areas of the development of cancer. And so this can be depicted kind of on this graph over time. You've got age on the x-axis and just sort of an intensity scale on the y-axis. And you can see that as you start aging, very quickly your stem cells stop dividing and your mutation rate jumps up. Okay, and as you live your life, you're accumulating mutations, and eventually you cross a threshold where your cancer incidence becomes more significant. The odds of you getting cancer are much higher. And so essentially what we're trying to do is understand how mutation rates accumulate in space and how we can potentially lower the mutation accumulation for astronauts and push back this cancer incidence rate to either be later in life or not at all. And so you might be wondering, why is this relationship not linear, right? Why does one mutation not equal one cancer? Well, first, we have this whole idea that you need multiple mutations in a single particular cell, but also there are things going on in the cell like DNA repair, which is something that I'm very excited and fascinated about, and I will tell you now. So if you're not technical, I'm very sorry. Hang on, this will be over in hopefully a minute or two. And if you are technical, hopefully this isn't too much. But when you experience a DNA double-strand break in cells, DNA repair can occur through several different mechanisms. One of the easiest way to explain is a repair function called non-homologous end joining, or NHEJ, where the cell will basically look at a break, send a bunch of proteins to grab the two ends and essentially join them together. This is a very error-prone process and leads to large deletions and mutations. A little bit harder to explain is homologous recombination. So this is a very accurate way that a cell can repair DNA. And essentially what it does is looks at other DNA in the cell that has the same sequence as where the break was and around the break, and it basically copies it. So it's kind of like, maybe back when you were in your chemistry 1 class and you didn't know the answer and you looked over to cheat off of your lab partner's paper. So the cell basically looks at a different copy of DNA that's identical, copies it down, and fixes the repair accurately. But this can only happen in very limited time points in a cell's life. And then we've got alternative end joining, which is basically somewhere in between, where cells can sort of look around and see chunks of DNA that look the same, but maybe the teacher's walking around and you don't want to look too long. And so it's kind of a hybrid of these two, where it copies a little bit and then it kind of rams some together. And this is also an inaccurate version of repair that results in small deletions and small mutations. Now, obviously, very fast and very oversimplified, but I think hopefully this helps you understand kind of where we're coming from. So why do we need to know this information? How can we use this information to prevent cancer in astronauts? Well, we want to help cells choose the right DNA repair pathway when they're hit with cosmic radiation, or at least block the pathways that are going to lead to more mutations, because more mutations equals more cancer. Okay. Luckily for us, several studies have shown that high energy particles hitting cells actually deactivate this NHEJ pathway, which is probably the biggest potential problem. And it just gets shut off in response to really high energy particles. We don't really know why, but we have seen over and over that that's the case. And so we're left with these two pathways: homologous recombination, accurate, but only a certain amount of time, and alternative end joining, inaccurate, and can be used at different phases as well. So most of the rest of the talk is going to be focused on alternative end joining and trying to block this pathway. And the way that we're going to do that is focus on the star of the show, a protein called polymerase theta, or POLQ for short. And so POLQ is the main effector protein that drives alternative end joining. So we're going to try to stop polymerase theta from functioning so we can stop alternative end joining and result in essentially only homologous recombination, an accurate form of repair for our astronauts. So the project that was recently funded by Axiom and others, we have this research question. If we inhibit polymerase theta, POLQ, can we reduce cancer risk and mutation rate following exposure to cosmic radiation? So a quick summary, if you zoned out for the last 5 minutes, cosmic rays plus alternative end joining equals high cancer risk, and cosmic rays minus alternative end joining, we hope, results in reduced cancer risk. So the way that we're going to test this idea is use a drug called ART558. It's a polymerase theta inhibitor. It has a very nice IC50, if that means anything to you. And we're excited to see whether this results in less mutations after cosmic radiation. So how are we going to do that? We are first going to perform a ground study using cells in a dish. So we've partnered with Columbia University. They have a radiation facility that can do full spectrum galactic cosmic ray simulation, which is a mouthful, but very cool to say. And they essentially are using a linear particle accelerator to do a multi-spectrum radiation beam that will mimic space radiation, and they've shown that it does that pretty reliably, similar to the NASA Space Radiation Lab at Brookhaven. So we'll take our cells, we'll have some that have no drug and some we will treat with our polymerase theta inhibitor, expose them to galactic cosmic ray simulation. And our readout for genomic integrity is threefold, and I'll get into this a little bit more, but essentially we'll look at cell survival, we'll take a look at large-scale chromosomes. So we're zooming in a little bit to look at the structure of the chromosomes, and then we'll zoom in even more and do whole genome sequencing to get down to an individual base pair level of resolution. So just a quick example for the non-biologists, what do these experiments look like in general? Cell survival assays look something like this. This does not belong to me, but I found it on the internet, and it's a great example. So this particular group is increasing the amount of radiation, 0 gray up to 8 gray, on these two different genetic conditions. And all the purple dots you see is a cluster of cells called a colony. And so you can count the number of colonies, and that's essentially, you're able to track cell survival in this way. And so you can see as radiation increases, the amount of purple dots decrease. And so the survival of these cells is decreasing. And in this particular example, they have some kind of genetic manipulation. They've knocked out something called MALAT1, and that results in more killing than cells that do not have that gene knocked out. So we're hoping to essentially, generate data of this nature using our drug rather than a genetic knockout. Another thing I'm very excited about is called chromosome painting or directional genomic hybridization. So we're working with a company called Chromatid Inc., and they do this directional genomic hybridization. And essentially what that means, they've got these chromosome paints where you can hybridize. It's essentially a whole genome fish where you're hybridizing tiny DNA probes all the way across a chromosome. And they can do this for all 23 chromosome pairs. And you can essentially measure whether there are any large-scale structural changes going on in the DNA as a result of radiation. So what does that mean? What does that look like? As an example, this was on chromosome 3, and you can see that the paint is continuous here, only broken by the centromere, which is normal. And in a damaged chromosome, you can see a particular lesion called an inversion. So you've got the paint continuous, and then all of a sudden it ends up on the other chromosome arm before reverting back. And so this is a particular type of damage that can be read out by chromosome painting, among many others. So we're very excited about this type of analysis. And finally, whole genome sequencing. We're very interested in looking at individual base pairs of the DNA in these cells and seeing what changes are happening. So we'll have of course, a reference genome for our cells of interest, the TK6 cells, and cells that get radiation plus and minus the drug. We'll have DNA sequencing information about what exactly is happening down to the base pair, which is really great high resolution data. And beyond that, we can also detect structural variants in a very similar way to the chromosome painting, but two methods always better than one, especially if they kind of work in a slightly different way that give you confidence that what you're seeing is reproducible. And so this whole genome sequencing can provide structural variation information on all sorts of damage like translocations, deletions, inversions, duplications, copy number variation, chromothripsis, all sorts of really interesting data that could come out of this. So we hope that our kind of multi-resolution readout will give us a really nice sense of what's going on in the cells. So what could be gained from this project? What's the whole point of this? Why have I gotten a little bit of money to try to do this? Well, we think that we're going to be able to determine if polymerase theta inhibition reduces cancer risk following exposure to cosmic radiation. But beyond that as well, this one particular drug is interesting, but we think that this methodology can be a really nice way to assess additional drugs that either work in the same pathway or different DNA repair pathways. Anything that can really modulate the genotoxic effect of the radiation, we think we would be able to read out using this methodology. We're hopeful to expand to some animal studies on Earth, on the space station. We'd love to do it on the moon. We'd love to do it. And overall, I think in general, we'll broaden our understanding of DNA repair in response to cosmic rays and radiation. So the roadmap here, our ground study that I just described to you is being done in human cells at basically the beginning of this next year, starting in the next couple of months. We're optimistic we can get, maybe on a small satellite, a CubeSat or something like that, either probably the end of 2024 is maybe too optimistic, maybe 2025. And we think that, when the Axiom station goes up, which is so far scheduled for 2026, we hope that we can run a trial on Axiom or any commercial space station. And we think that, we could be fully operational for Lunar Gateway and Artemis 4 by 2028, which is just the timeline that NASA has set currently for Lunar Gateway, which may, slip into the future. So yeah, our team is based out of Memorial Sloan Kettering, led by myself and Dr. Daniel Higginson, who's a radiation oncologist there. We've got funding from a bunch of different places, and we're really excited to do this work. We've got our critical assets in particular. Working with Columbia is going to be excellent. Chromatid, all of the great facilities at Memorial Sloan Kettering. And so we're hoping that this project, at least the early stages of it, can be done by summer 2024. And yeah, that's been the talk. If you enjoyed the talk, I just want to plug a couple of other people. I don't get any sort of monetary benefit from this. I don't even think they know that I'm doing this. But if you enjoyed this talk or this topic or anything about space, biology, etc., I would love if you checked out Chris Mason. Dr. Chris Mason's at Weill Cornell. And he just published a fantastic book called The Next 500 Years, Engineering Life to Reach New Worlds, all about genetic engineering of humans and bacteria to try to help us get to other planets. Dr. Erica Hamden, the University of Arizona, I had the pleasure of meeting this past summer. She gives a great YouTube talk, What It Takes to Launch a Telescope. It's basically about resilience in science and what happens when things inevitably fail. Dr. Beth Shapiro at UC Santa Cruz has written a book and given plenty of excellent lectures about how to clone a mammoth and the science of de-extinction. Spoiler alert, we're not very close. And if you enjoyed the talk, please check out my LinkedIn here, giving you a QR code to scan and check out. And yeah, thank you very much. I want to thank Memorial Sloan Kettering for sponsoring the work, as well as Boryung, Axiom, the Aurelia Institute, the Humans in Space program, and of course the AIAA for hosting me. So thank you, and I'll take any questions.
Fantastic, thank you, Dr. Levy. Any question here in the attendee or online? I think John, Mr. Berza asked a question. So John, do you want to speak out? You have the microphone.
Yeah, so could you just discuss the potential, does this drug potentially lessen the destruction from both cosmic solar radiation and galactic space radiation? And what's the What is the differences between them?
Yeah, sure. It's thank you for the question. I think in general, so I'm much more of a cell biologist, but my rough understanding of the difference between the two is that there's a maybe I'm not sure how large, but there is a large difference in terms of the energy between them. So my understanding is that solar radiation is more easily shielded against, and galactic cosmic rays are a much higher energy, more difficult to shield against. And so there are some pharmacological interventions that we think will work. And on that end, while I can talk about it, why don't we just use shielding? I think let me try to move this. Is a reasonable question. As I'm saying, solar radiation is easier to shield. So it turns out cosmic rays, since they're so high energy, not very easy to shield. So I've read in different places, you need about 13 feet of lead to shield from a cosmic ray, or about a swimming pool full of water. Neither of those particularly easy to launch into space. And one problem as well with cosmic rays, I couldn't find a perfect image depicting this, but essentially if you don't shield a cosmic ray completely, what happens as it hits Earth's atmosphere or your shielding of choice, it will fragment into basically many versions of secondary radiation, almost like a shotgun shell. So anything behind that shielding is actually going to experience more radiation by surface area than you would otherwise. And so, yeah, it's a good question. And we think the drug will primarily function on galactic cosmic rays.
Excellent question. I think Richard raised hand. Richard, go ahead.
Yeah, thanks, Devin. That you just explained really what I was going to ask, because you don't really get GCRs until you get outside the Van Allen belt. I mean, the Apollo astronauts did have issues with flashes in their eye, in their retina, and I think potential lesions on their brains that took place from these GCRs hitting them. But I'm wondering if you have a spacecraft that's aluminum outer shell, you're possibly going to get that spallation anyway before it gets inside the living accommodation. And I know that there are people working on it. Some people have been working with it in the past. So is the damage, if you get a spallated GCR, is the damage similar than if you just get a solid whack from an ion?
Yeah, it's a good question for sure. I think I don't know that I don't know with total confidence, but I would say essentially the spallated the secondary radiation would look more like a gamma ray. So those particles are slowed by shielding, even though it's insufficient. The problem I think is that I think you'll receive less damage in a singular instance, but because of the spreading out of those particles, more cells are going to be affected, and so it's. I'm not quite sure how the math works out, but more mutations equals more cancer in my silly little saying. And so I think more cells getting hit with more particles actually could be worse, potentially, but I don't have any data for that.
Thanks.
Thank you, Richard. I think Pedro Silverman, Mr. Silverman, you want to speak out?
Sure. So I was wondering, for the CubeSat experiment, will you be able to get enough radiation for you to see an effect on the cells? Because it might be kind of hard to have the human cells last for more than a couple days in orbit.
Yeah, it's a great question, and thank you, Dr. Silverman. Good to see you. The tracks that you see here, I think this experiment is done on a beam, but there are experiments that were done on near using cells, and they see these same radiation tracks from GCRs. So I think I think the quick answer is yes. I think we would see the level of damage that we would need. And Dr. Erica Hamden is an astrophysicist who launched the Fireball satellite, and she published a paper that talks about basically how many cosmic rays were hitting her telescope, the Fireball telescope. And I forget the exact number. I'll send you the paper later, but it was quite high. So I think I think it's an odds game, but I think that we would see sufficient damage. I mean, I hope that we do.
Very good, Rich. Actually, since Richard here, actually, I want to echo some of the other questions he mentioned. Richard mentioned about the Dream Chaser, right? Dream Chaser. The Sierra Nevada Dream Chaser as a good launch vehicle for possible biomedicine experiment. I'm speaking, actually, you see our section is called Los Angeles and Las Vegas section. So actually, our Las Vegas chapter people, Mr. Marty Waltman has been working with the project of the Dream Chaser to be launched from Florida or Vandenberg and landed in the Las Vegas airport, McCarran or Harry Reid Airport. So that's an interesting possibility. Of course, his focus is more on space tourism. But actually, it could be a good potential. So if you like to work with our AIAA Las Vegas chapter for this possibility, we'll be happy to make a connection. Okay. Yeah. Right. Thank you. And the other thing, you know, actually, many years ago, I used to work in a cancer center to support biophysics study, and they actually had also. I don't remember the detail, but some of the researcher was also talking about this counter radiation drugs. So for you just mentioned it's for different kind of radiation environment. So how fast it is? Is it kind of immediate remedy, or is for like a vaccine for prevention? What is the vaccine that you are working on? Is it for like injection or pill, or is for that vaccination, or is it meant for treatment?
Yeah, it's a good question. We don't have all of the details worked out. So the drug is currently in a phase 1 clinical trial for actually radiosensitization of tumors that have defects in homologous recombination, that pathway that we're trying to increase and support. And so my understanding is that it's a pill that you would take basically before radiation exposure, so that the cells are sort of primed to make that decision once they're exposed to the cosmic radiation. There's all sorts of questions that we have about that, including, you know, how many pills would you need to take? How many do you need to bring with you? Does the cosmic radiation itself impact the pills, and how? So, you know, we're excited to do the ground study and take this first step and just show that the drug has a radioprotective effect in this context. And then, you know, in time we'll we'll take the next step. And part of that is getting a pharmacist on board and trying to understand some of the finer details of that, because I don't have any sort of pharmacy experience.
Excellent. So we might have some more questions here, but we have attendee here right now. Okay, what happened is I think we're supposed to have a 4th speaker, Kimberly, but she cannot make it, and she belongs to also a group called the Girls for Space. And right now is from that organization, so she would like to kind of tell us briefly about herself and this organization. So right now, go ahead. You can unmute your mic or even turn on your video if you like. I guess it is not ready yet.
Well, I have a question while we're waiting, if that's okay.
Go ahead.
Actually, a couple of them. This is Martin McLaughlin again. First of all, congratulations. This galactic cosmic ray issue has been swept under the rug, in my opinion, too long. We all are confident that we can shield against solar particle events, but you know, a proton going at near the speed of light is a tough animal to deal with. So good. 2 questions. I've heard about some work on doing selection of astronauts. That there's some variation, I don't know how wide, in the susceptibility of individuals. Is there anything you know of in that area to take that? And my second question is, I've also heard work on, well, let them get exposed and we get them back, we'll fix them with bone marrow transplants or something. So those are my 2 questions.
Sure, those are great and interesting questions. Particularly the second one, I think that's interesting. Yeah, for the first question, is there any sort of predisposition for, I guess, for cancer? Certainly, I mean, you know, genetically there are all kinds of predispositions. I think I would love to know how astronaut selection takes place. I think that's been shielded for a long time, and is I think even you can't do a freedom of information request, although some have tried. So it would be interesting to see if they're doing something like whole genome sequencing on maybe the final few candidates or something, and you know that field itself is kind of, it's, you know, a little bit wobbly. Like how can you really know? It's almost like Minority Report. You know, how do you know that some particular DNA variants will lead to increased susceptibility? It's not always so clear. So I'm not, you know, I'm not really sure for that question. The second question I think is interesting too. Should we just let the, you know, maybe that's more of a moral question, or more of a question of consent for particular astronauts. Do you just let them get exposed to the radiation and roll the dice and try to fix it later? You know, I don't know the answer to that, and I don't know the morals of that. I'm sure there are people much smarter than me who will go into the bioethics of that. Personally, I would not want to do that, especially if something like this exists. So currently there are no countermeasures to GCR. So if this exists, I personally would want to take it rather than take the risk of getting cancer and getting a bone marrow transplant. I think that's much more traumatic than my drug, but but I also have to prove that it works. So
That's great. Thank you.
Yes, you're right, because I think both from your research, and Alina also mentioned this organelle. This things regarding you know, the stem cells, you know, or those kind of experiments, or those kind of ethical issues. So this is something that yeah, we have our technical chair, Gary, Mr. Gary Moyer here. So Gary, do you want to say a few words? Oh,
Actually,
yeah, you need a microphone. You need a microphone.
Right. Where is that? Oh. Yeah, hello, this is Gary Moyer. I had quite a diverse career over the course of my many years in the industry. One of the interesting things I did was to work on a system called the attached shuttle payloads, which was actually a PRW division which was created specifically for that purpose and to minimize the cost of figurehead. Anyway, I came into that group when they presented the problem structurally. And in the course of an initial assessment, I could not identify the structure that supported these heavy test specimens. We ended up with a crew of 4 people repairing that configuration over the course of about 6 months. But that configuration was designed to grow sodium iodide and other exotic crystals in a weightless environment. And so the question that I really would like to ask is, did you discuss the structural and the services necessary from the space station to support this system? I'll pass the question back over to you.
sure. So sorry, just it's a little difficult to hear. So to understand, just to recap the question, have I clarified with NASA the types of structural support needed to do the experiment? Is that what you're asking?
Yes, but also in addition to structural, it's also the, you know, all of the power, the expendables.
Sure.
Are necessary in the face.
Yeah, sure. Yeah. Okay. So good question. I thankfully I am happy to, again, kind of kick the can to people much smarter than me for things like payload integration and things of that nature. So I know companies such as SpaceBox, who presented here today, as well as many other companies, NanoRacks, I think, plenty of sort of payload launch providers kind of handle that end of it. So I am lucky enough that I just get to be on the scientific side and try to come up with cool ideas and try to win money. And when I win the money, I get to go to these companies and say, hey, I want to do this experiment. I need you to make sure that it works structurally and power and all that stuff. And they kind of work it out. And there's always a negotiation. But yeah, I think, experiments like this have been done for a long time, and there's nothing too wild about these requirements for power or anything.
What kind of geometry or what kind of interfaces or volume and weight do you need to do your tasks?
Yeah, so the project we have now is a ground study, so we can luckily do as much as we want, but we were looking at CubeSat size volume. It doesn't require too much of a footprint. Yeah, I think something that, if we were able to get on ISS or Axiom, we could use one of the NanoRacks type footprints. So they've got plenty of variable sized racks that are basically cell culture incubators. Hopefully that answers your question. I'm not too much of an expert on that end of things.
So you're actually saying that you can actually use in the CubeSat kind of volume?
Yeah. Yeah.
Okay. The ones that I worked on were roughly a little over a foot cube, and they included all the support systems necessary in the specimen test elements, and they were supported in the structural rack. That was partly what I worked on.
Mm.
But those cubes were on the order of 50 to 70 pounds.
Mm-hmm.
So you can understand that when the structure came in looking something like a trailer's, like a house trailer's structural supports, but that wasn't going to carry it for launch and landing possibility. So
yeah, I can imagine that would be a big problem.
CubeSat, that's an easily solved support problem. Although if you still may need interfaces for power and even expendables. CubeSats are pretty limited in capabilities, but I'll go one other step in the process in my career. When I hired into TRW, one of the projects I was hired for was the Viking biology instrument. And so that little 1 cubic foot, actually, it was a little smaller than that. Laboratory actually supported 3 independent laboratories, plus an additional, basically CubeSat size specimen area that carried the support materials such as gases and other nutrients and so forth that were used in the other 3 experiment labs. So that was a watchworks. It really truly was. And they also had another cubic foot that was used for the electronic support and computers and so forth. So I didn't work on that part. But at that point, I kind of ran out of my question. But you do operate in a 1 atmosphere environment, basically like the space station, or do you want, or are you happy to operate in an unmanned satellite?
Yeah, I think I think 1 atmosphere would be great. I mean, the cells have to live and grow and survive. So yeah, they need to be, atmosphere, like as though it were on the ISS, particularly if the drug is meant to intervene on something like ISS or Lunar Gateway. It would be good to have the environment be as close to that as possible.
Yeah, I indeed ran out of my question, but.
Yeah, you can, if you have more questions, we can see, right now, Red Hawk, do you want to speak out? You have the mic.
Yes.
Yeah, just say a few words about you and your goals for space.
Yeah, sure. First of all, hi everyone. My name is Ragat. I am from Jordan, and I'm part of Space for Girls. Actually, I'm a mechatronics engineering student in my 4th year, and interested in space in general, specifically medicine in space and rovers. Our mission in Space for Girls is to empower young women to become innovators in the field of STEM and to become the next generation of leaders in the rapidly growing space industry. Specifically, I'm a part of a program called Stellar Rising Stars. It's a program to immersive and educational initiative tailored for skills women, selected, sorry, for selected women ages from 17 to 25, and aims to break down barriers and stereotypes by encouraging young women throughout the world to confidently pursue their interests in space-related fields. So this is about Space for Girls. Let me thank you for this really inspiring and amazing work, and hope and wish the best for you.
Thank you, Rebecca. Thank you. Stay in touch. Actually, I do have, if nobody has any more questions, I do have a quick question, because obviously, Yaroslav introduced you. So are you working with SpaceBox, or you plan to have something working with them?
Yeah, so I had the pleasure of meeting SpaceBox, Yaroslav and Alina at the Ascend meeting in Las Vegas. So we were all we all competed, although fortunately they were on one competition and I was in kind of a separate competition. So we were not competitors with one another. But we met each other and got to know each other a little bit, and are excited about the possibilities in front of us. So I think they're doing really interesting work, and there's clearly, I think, an overlap between scientists and technology providers and launch providers, and the more of us we can get into a room, the better the odds are that we can get some really interesting science and data to move forward. I think it will take everyone. Every mission takes hundreds of people. Everyone has expertise in different things. So it's been really great to get to know them, and we wouldn't have met if it had not been for the AIAA and for Ascend. So thank you. I know this is the, I guess, LA Las Vegas chapter specifically, but thank you to the AIAA team more broadly as well, and for having us today.
Yeah, thank you. Well, I didn't realize you were there. Actually, the Axiom Space, if you and Richard talk about Axiom Space, actually the VP, former VP and chief scientist, she was there. I don't think we get a chance to meet her. She was there, and that's a good thing for Ascend. And next year is going to be in Las Vegas again. All right, Deborah, thank you. Stay in touch. So that's really good Axiom Space. Actually, just like Gary, he used to work in Viking on the first Viking and actually some of our members like Alicia, let's see, stay a little bit, please, you're welcome to speak up. Basically, I wanted to say, do you know that you also mentioned about Artemis 4? But actually, this December, I believe this Artemis I think this company called Astrobotics in Pittsburgh, Pennsylvania, they are going to launch. They descended to Florida. They are going to launch the Peregrine lander, the robot, to the moon. That's the first American lander back to the moon since Apollo 17. So it's this December, as early as this December. So that's another company. You talk about space station, but AIAA has some members. They actually working on sending directly to the moon. That's something could be interesting. And eventually the goal is, I understand the space station is kind of like a national laboratory, but eventually people are going to the moon and Mars. So you could consider maybe doing something, on the moon or Mars, even Mars. And we can see how this Peregrine launch and also Elon Musk Starship test open. That's probably going to do, yeah. So just some comment. So Alicia, do you want to say something? I think you are talking about, mention something in the chat. If I'll give you the mic. If you want to speak up, please. If not, then I don't see any more question here. So Alicia, you have the mic.
Hi, sorry, I do have to hop off in like a minute, but I just wanted to put in the chat, just to introduce myself. This is a really great couple of talks. I'm Alicia. I'm the founder of A Billion Dreams, a nonprofit, and we engrave names and dreams of kids, folks going through cancer treatments, and loved ones on space missions. So our first one is launching on a plaque on one of the SpaceX Transporter 10 missions. I'm a young cancer survivor myself, and I put my LinkedIn in the chat for anyone who would like to connect. And I'm really excited to keep learning more about folks' research. And this is my first AIAA event, so hopefully the first of many that I get to be involved with.
I had one more technical question. How quickly do you need to progress from cellular to bigger models, mice, humans? And what's that roadmap look like? And what kind of durations do you need for those experiments?
Yeah, that's a great question as well. Thank you. So we're starting in cells. Obviously, it's a big leap, as it always is for any drug, to go from cells to humans. We're interested in partnering with the Trish Institute, Translational Research in Space Health, I think is the acronym, out of the Baylor College of Medicine. And so they have we're going to write some grant work for them for this project, and they really are not interested in animal model data. They want to see implementation of a potential countermeasure within 3 years or so. And so we're trying to stick to that timeline to jump basically from cells to humans in about 3 years, which seems a little ridiculous. But I think one thing that's working in our favor is that the drug we're proposing to use is under a clinical trial, phase 1, for human safety for a different purpose. And so that data is set to come out in 2025, and we're not involved in that study, but we will benefit greatly from seeing that humans can tolerate the drug and there are not any crazy side effects or anything. So the roadmap is not fully clear, but the Trish Institute has been very clear that they do not want animal data. And so we're working towards trying to achieve that. So we're hopeful that, the teams running the clinical trials, that things go well and that we can kind of continue to work up our data, but it's always, you saw Alina's funnel of kind of drugs that, don't make it through the valley of death. So of course, kind of going from a cell anywhere is really tough. And so, we'll see what that looks like, but we're optimistic. That's all we have is our optimism, I guess.
Excellent.
Hopefully that answers, yeah.
Any question? Any more question? Okay, if not, let's thank Dr. Devon Levine the team this fantastic presentation. Thank you so much. And
Thank you for having me.
And Alina, very good. So appreciate it. And all the people online, I know you don't want to come here, but hopefully we can see you in person pretty soon. Thank you.
Yes, I look forward to seeing you in person, hopefully next time.
Unfortunately, after pandemic, a lot of people prefer to stay online so we can see what happened today. It's hard to get people out, but we're gradually moving there. More and more people showing up. Okay, thank you. Have a wonderful day. Stay in touch.